Other videos this week have focused on how DNA is used to build proteins, right? First, that process of transcription, DNA to mRNA, and then that mRNA is translated into a protein, that physical expression of that gene. But even though all of our cells have the exact same DNA, the exact same set of DNA, these cells in your fingernails have the exact same sequence of DNA as these cells in your stomach. But they're very different cells, right? And they're doing very different things. So how do we get those differences in cells, in their structure and their function, if they have the same set of instructions? Through gene regulation. Not all genes will be turned on at all times. And differences in which genes are turned on in which cells give us the specialization of cells. We use the term gene expression to describe the process of a gene being turned into its physical product, the instructions in that gene being used to build the physical product, right? Through the process of transcription and then translation. But again, we don't need all of the genes in all cells active all the time. Some genes might only be active early on in childhood or during adolescence, or maybe they get activated during aging, right? And we don't want, for instance, the gene for how to make pepsin, how to create the enzyme that can break down proteins, active in cells besides really our stomach cells. That way they can secrete it into the stomach, right? We don't need the gene for producing light-sensitive proteins anywhere besides our eyes. So genes will be regulated, turned on or turned off in different ways. So that way we're not constantly making a bunch of proteins that are not necessary for the cell. Let's start first looking at how this works in prokaryotic cells. This process of prokaryotic gene regulation is somewhat similar. We don't really see this in eukaryotes, but it's a good example of how the environment and feedback from other cells can alter gene expression. So prokaryotes organize their genes into segments that we call operons. So operons is just several genes that are all involved in producing a similar product. Here, we are looking at what's called the lac operon, which are all genes associated with producing the enzyme lactase. And lactase is an enzyme that breaks down lactose, the sugar that's in milk, right? So lactase is the enzyme. And this is what these genes here are doing. These genes contain the information for how to build that lactase enzyme. So, all of these genes are arranged in basically the same location. They're all next to each other in this region of the DNA of a prokaryote that we call an operon. Remember we talked about there's a promoter region? And that promoter region is where RNA polymerase will bind to begin the process of transcription. In prokaryotes, we have some extra pieces before we actually get to the genes that contain the information for how to make lactase, these genes right here. There's a little segment of DNA right here that we call the operator. Okay, this is all DNA. All of this strand right here is just DNA. So the promoter is where RNA polymerase will bind to begin transcription. This piece of DNA right here, this that we call the operator, acts like a switch to either allow RNA polymerase through to transcribe these genes, or it can physically block RNA polymerase from going through. Why would we want to physically block RNA polymerase from transcribing these genes? They're just lactase enzymes, right? Well, building enzymes takes energy and it takes amino acids, right? What if there's no lactose around in the cell? Then if there's no lactose, this enzyme would be useless. We don't need it right now, right? So what happens This is super cool. What happens is that when there is no lactose available to this prokaryote, this molecule right here called a repressor will be bound to the region of DNA that we call the operator. Remember, the operator just acts like a switch, turning on or off this gene. As the repressor is bound to that operator, it's physically blocking RNA polymerase from transcribing that gene. And the repressor will only be bound to the operator when lactose is not present. So no lactose, this gene is off, which is fine because we don't need these enzymes right now if there's no lactose around. Because these enzymes, again, lactase breaks down lactose. Okay. But what if lactose is in the cell? Let's say there's an influx of lactose. This cell now has that available as a food source. These enzymes would then be useful when lactose is around, right? So what happens when lactose is available? That lactose will bind to the repressor. And remember, when anything binds to a protein, it causes a shape change. So when lactose binds to that repressor, that causes that repressor to fall off of the operator. So the repressor is no longer bound to the operator when lactose is available, when lactose is inside the cell. Now with that repressor gone, RNA polymerase can just plow right on through, can go right on through the operator to transcribe LAC-Z, LAC-Y, LAC-A, create that RNA transcript, which will get turned into the protein of different lactase enzymes. Pretty cool. Kind of a simple, elegant solution to this problem of we don't want all genes present and working all the time, right? So RNA polymerase can move through and make those genes, make these transcriptions of these genes right here to make lactase enzyme, as long as lactose is present. But as soon as the cell starts to digest this lactose, as soon as these enzymes start to get to work and break down this lactose, and that concentration of lactose drops, this repressor will then unbind from that lactose and reattach to the operator. And we're back to being in this scenario up here, where the repressor is bound to the operator, which means there's no lactose available, which means we don't need these genes. So the repressor is going to prevent RNA polymerase from transcribing these lactase genes. Gene expression in eukaryotes is a lot more complicated. There's a lot more going on in eukaryotes. We have a lot more DNA, we have a lot more genes in general than prokaryotes. So we can regulate gene expression at many steps along the process of DNA to RNA to translation. There's many different layers to gene expression or regulating gene expression in eukaryotes. We can regulate, remember we talked about how coiled up DNA can get? Some regions of DNA will be more open, less coiled up, less compacted, and some regions of DNA will be more compacted. So we can actually alter how unwound or wound up the DNA is, and that can affect gene expression. And we do that by altering those histone proteins which DNA is wrapped around. We can add in things or take away things called transcription factors. Transcription factors are kind of like that repressor, but they aid in regulating genes, in which genes are turned on or turned off. Remember, if I say a gene is turned on, I mean it's being transcribed and translated. So hormones often will act like transcription factors that will bind to the DNA and enhance or silence the transcription of that gene. Okay? A lot of signaling molecules that cells will use to talk to each other will often work as transcription factors, or their end result of that signal will activate a transcription factor, which will alter which genes are being transcribed or not transcribed. We can also regulate that RNA processing, right? So even if a gene is transcribed into RNA, we can regulate the process of removing those introns, splicing together those exons. We can either speed that up, slow that down, or we can change which exons are left in there. It's a whole other layer. We can get many different proteins from one gene by altering the combination of exons that are left in that mRNA. We can regulate translation. There's ways to flag certain strands of mRNA for destruction by the cell, so that way they won't be translated, right? So we can say, this piece of mRNA, we don't need it anymore. Other molecules will bind to that mRNA and destroy it, basically. So we can regulate the process of translation. We can regulate protein activation or folding, right? Remember we talked about coenzymes and cofactors or protein activation? Even if we have a protein made, if it's not folded properly by other proteins, or if we don't give it an activator to work properly, then that protein won't function, right? So there's many different layers in which eukaryotes can alter the gene expression, but the key idea that I would like you to take home from this is that not all cells need all of their genes functioning at one time. In different environmental factors, as well as different signals from cells, whether they be hormones or maybe a neurotransmitter, will alter which genes are turned on, meaning transcribed and translated, and which genes are turned off, are not currently being used from the cell. There's a whole wide world. People spend their entire careers studying certain transcription factors or how certain genes in eukaryotes are regulated. This is a really, really complex process. Really interesting, crazy interesting. But there's a lot going on here. I'm not going to expect us to go into the details. I just want us to know that there's many different layers, different locations along this process in which genes can be regulated.